Hello students, welcome to NCERT Solutions by Vedantu. I am Ms. Gopika, your own biology master teacher. So today we are going to do the chapter Microbes and Human Welfare. And yes, awesome, we are live as well. Let me also check the voice once. So today we are going to... Yes, awesome. We are live. So I hope all of you have been enjoying our NCERT line by line, and Baswa sir, Diksha ma'am, myself, Navamita ma'am, Namrita ma'am. So we are a group of teachers who have been teaching you NCERT line by line from past few days, and I hope all of you are really enjoying it. So today we are going to do microbes and human welfare, which is not a very it's not at all a difficult chapter, but you can expect good amount of questions from this chapter for the main reason that there's a lot of terminologies to remember. Okay, so today we will do NCERT line by line and try to understand microbes and human welfare. So let's directly dive into the video. So students, let's start with our topic. Okay, so microbes and human welfare. So every year, usually NEET, we tell the questions are around 2 questions. That means a weightage of 8 marks we can expect for the NEET students, and for the board students it totally depends on your syllabus, right? If you have that topic, then definitely more questions are going to come from these topics. Okay, chalo. Now all of you, do not forget to subscribe to the channel and also hit the like button so that we know, and that's our encouragement that you can give to all of us. So let's read every line of NCERT. Besides macroscopic plants and animals, microbes are a major components of biological system on earth. You have studied about the diversity of living organism in class 11. Do you remember which kingdom among the living organisms contain microorganisms? So all of you, I have only taught biological classifications for all of you, and in biological classification we have learned, okay, one second, in biological classification we have learned a kingdom called as kingdom Monera. Kingdom Monera. And this kingdom Monera sole members were none other than your bacteria, right? Sole members were microscopic bacteria. And then in the last, at the end of the chapter, we learned about few people like virus, viroids, prions, all these people who did not have a place to be. Now imagine you do not have a place to stay, right? You are not having a place to stay. That means we will put you somewhere, right? So same way, these people were not falling under any classification, which was like, okay, Whittaker was not ready to put them under any classification because of the reason that these people are not belonging, or like they are not able to have common characters with some other organisms. Okay, so virus, viroids, prions we learned, and we understood that kingdom Monera sole members of bacteria. Okay, so this is the question. Now students, every time like human health and diseases, that chapter, just imagine I will always teach you about the problem causing people, right? The pathogens. See, there might be a set of people who are causing troubles in the society, and there will be another side of people who are really good. But what happens is, when we talk as a whole, we'll be like, oh my god, bacteria is so bad here. Bacteria is very, very bad. Okay, bacteria will always cause a disease. So this way, what exactly happens is, we are making the entire bacteria or the entire microbes negative. So this chapter is going to show you that microbes are actually very, very helpful to us. There are good microbes and bad microbes. Same way in our body also, there is good bacteria, bad bacteria. So we should understand the fact that not everything is bad. Okay, not everything is bad. There are the good ones. Okay, so yes, you should be the good ones. Okay, all of you should be the good ones. Yes, it's clear. Okay, chalo. So students, which are the ones that are only microscopic? So we spoke about it. Microbes are present everywhere in the soil, water, air, inside our bodies, and that of other animals and plants. So now imagine, let's take this pen. Okay, if I go and check it under the microscope, I will be able to see a millions of bacteria, for a very simple reason that my hands are filled with microbes. Okay, my whatever I'm touching, everything has microbes. Everywhere around me there is microbe, including in the air. Now how do I know, ma'am, microbes isn't in the air or not? How can you tell me like that? Very simple students, you take a Petri plate. Okay, I think all of you would have seen a Petri plate, right? This is how a Petri plate looks like. Okay, I think all of you are not marking your attendance. Quickly like the video. Okay, this is your Petri plate. In this Petri plate, oh, I'm so sorry. Yeah, in this Petri plate, let's say you grow a nutrient medium. Okay, nutrient medium means an agar which has all the nutrient content that is required for this bacteria to grow. And then imagine you open this nutrient plate like this. Okay, you just show it around the air like for 2-3 maybe a minute or so. You show it like this and you close it, and after 48 hours you just come back to the lab and check or look at this Petri plate. You will be amazed to see that there are colonies of bacteria. Okay, there could be colonies of fungi in that Petri plate. So this gives us an idea that everywhere there is microorganisms. We are not able to see them, but that does not mean they do not exist. Okay, now they are present even at the site where no other they are present at a site where no other life forms could possibly exist. Okay, sites such as deep inside a glacier. Students, it's a thermal vent. The temperature is going to be so high as 100 degrees Celsius. Okay, it's like boiling water temperature. So can you put your hand inside when the water is boiling? You cannot do that, right? So it is going to be that much temperature, okay, heat. Still these microorganisms will be able to survive. Okay, so these people are present everywhere where other human life is not possible. Okay, deep inside the soil, under the layers of snow, highly acidic environment. They could be present where it is highly salty solution, highly temperature acidic solution. All these places they'll be present, including inside our body they are present. Okay, now microbes are diverse protozoas, bacteria, fungi, microscopic animals, and plant viruses, viroids, also prions that are proteinaciously infectious agents. Okay, proteinaceous infections means they have a protein code outside, right? A bacteriophage is always what I draw. This is the protein code, and inside there is going to be a genetic material. So they are proteinaceous infectious materials. Okay, infectious people. Now just look here, you have some diagrams of your NCERT which is very, very important for you. Okay, look at these diagrams. Okay, here you have a rod shaped bacteria. A rod shaped bacteria. Rod shape means what? Like this. Okay, we call this rod shaped bacteria bacillus. Okay, or bacilli we tell, no, bacillus in the terms of your bacteria. So when we teach you biological classification, when I taught you biological classification, there was a diagram given below telling that on basis of shape we can divide bacteria into 4 types. One is rod shape, one is circular shape, one is thread like or spiralum, and one is comma shape. So we have bacillus, vibrio, spiralum, and your which is one more, did I miss? Coccus. Okay, coccus. So 4 types we have. Okay, 4 types we have. So this is the rod shaped one, this is the coccus one, matlab circular. Okay, coccus means what? Circular, ma'am. Okay, or we tell spherical. Okay, spherical. Now if I magnify this fellow, okay, if I magnify this fellow at 50,000 x magnification, okay, at 50,000 x magnification, this is going to get magnified to look like this. So it is going to be a rod shaped bacteria. Okay, a rod shaped bacteria with flagella. Okay, what is this flagella going to do? Flagella is going to do the job of locomotion. No, flagella is going to help them in slight kuti movements. Okay, kuti movements. Hi Tanuja, hi Himanshu. Yes, now we spoke about virons, prions, all of this, no, proteinaceous infections. What are those? See students, this is the outer coat. This is the head. Okay, this is the head, this is the collar, this is the tail, and this looks like they're like the legs, no, like that. But see, when you look at this, you'll be like, my god, ma'am, it looks like one electrical or one engineer built this virus, but it's all creations. No, so students, this protein code, inside this protein code there is going to be the DNA material. Okay, inside the protein code, who is going to be there? The DNA material is going to be there. To protect this genetic material, they have a protein code like this. Okay, and this protein is made up of what? Capsomeres. Individual capsomeres together forms capsid. So this is everything that you have learned in biological classification. A student who is not able to understand That means you haven't seen biological classification. Okay, you have to go back and watch biological classification. So I think diagrams are clear, no? Now, this is what I told you. See, first technique that you're seeing here is called striking. Okay, so here students, what we do is we make streaks. Okay, we make streaks, and these like we have different techniques to do this. Okay, different techniques to do this, and this is a fungal colony. Okay, this is fungus. Now, how did this people grow? See, this is different technique. This is how, very simple. Now, let's say Tanuja has got some bacteria. Okay, she is like, I have bacillus in my, with me. This is the bacteria. Okay, now I have a Petri plate. Okay, let's say Himanshu has a Petri plate. He has put the nutrient medium in this and he has kept it. Okay, now what is he going to do? He's going to take a circular structure like this, like a glass rod. He's going to touch, just touch. Okay, he's not going to dip and be like, oh, did you guys mixed? Are you all fine? They're not going to do that. Okay, he's just going to touch and he's going to start streaking. Okay, he's going to start streaking. Now, wherever he made the streaks, okay, next day when he comes, he will start noticing colonies in that portion. Okay, he'll start noticing colonies in that portion. Understood? So if you dip and you mix it like, oh, khichdi sambar you're making, no, then gone. Okay, you just have to touch it, because students, if you mix it, there's a lot of bacteria content, and other than the loop, there is a glass rod like this. Okay, there's a loop like this. Okay, there's a loop like this. Now in this loop, how much should be there? That much only should be there. Not like till here, if you have bacteria, you dipped it, you're like mixing it. If you do that, the whole glass plate will have bacteria. Then contamination will happen. Okay, so instead of that, in this loop only bacteria should be there. With that, you have to do streaking. Okay, like this you have to do, and the colonies will start growing maybe after 48 hours. Okay, depends. Bacteria to bacteria, changes will happen. Now fungal. Now Tanuja is saying, no ma'am, I don't want to see bacteria. I want to see if fungus growth is going to happen. What she can do is, she can just take a Petri plate, as I told you before. She can just take a Petri plate and open the Petri plate and just show it to the air. Okay, show it to the air, or go to areas where there's a lot of waste materials. Okay, where there is a garbage dump or a sewage area. Just show this material, and what happens immediately, students? Spores will be there. No, fungus will have spores. Okay, fungus will have spores. So those spores will automatically come and follow in this, and because we gave a nutrient medium, an agar, which is going to allow them to grow, they will obviously happily grow. Yes or no? They will happily grow then. So they will be like, okay, fine, I'm anyway getting a medium. It's better than, struggling. Imagine someone adopts you and they are very rich. Okay, you will jump and go, no? Like that only. This people's nutrient medium is like where we are giving them everything. So the spores will come and fall. And when Tanuja comes back, maybe less than 48 hours it takes, she will see like this green color patches. Green color. Sometimes it can also be darkish, dark green. Okay, dark green, almost blackish color. Okay, she'll notice. That's when she'll realize, oh my god, everywhere there is fungus also. Okay, this is your fungal colony. Fungal colony. Got it? Got it? You have sent a salad for the emoji. Okay, Tanuja has sent a salad instead of a fungal colony. Okay, so understood? This is fungal colony. How can you connect fungus with food? Okay. Okay, next thing, students, microbes in household products. So here you have to study some household, some medical, some sewage treatment, and some biogas production. That's all. This chapter will be over. Okay. So you would be surprised. Okay. You would be surprised to know that we use microbes or products derived from them every day. Okay, I don't know how many of you know this, but as I told you, when we always talk about the negative, we always tend to look at the negative. What happens? We only listen to the negatives, right? Wow, what a beautiful physiology. Sorry, students. Okay, so what exactly happens is, students, microbes are very, very beneficial to us. Okay, microbes are very, very beneficial to us, but the thing is, we do not know it. So if for day-to-day activities, we are using these microbes. Okay, for day-to-day activities, we are using this microbe. A common example is the production of curd from milk. How many of you in the class have ever heard your mom telling that, take 1 spoon of curd Okay, add it into the boiled or the lukewarm milk that I've kept near the kitchen. Okay, she might have taken the milk, maybe one glass only, not like a tumbler or anything. One putty glass she would have taken. In that she would have boiled the milk and she would have kept. Okay, maybe like half or this much. Now what you are going to do, you are going to take curd, 1 spoon of curd, and add it into it. Okay, Tanuja, after the class, if you tell me, ma'am, I'm feeling hungry after sending all the food emoji, then it's there for you. Okay, so what is going to happen? So you take 1 spoon of the inoculum and you add it into the milk. Now, this inoculum that your mom asked you to add and you forgot, or maybe you got hittings for, is full of lactobacillus bacteria. What is it? Lactobacillus. Okay, lactobacillus bacteria. Understood? Lab, lab. Lactobacillus. So the inoculum that your mother asked you to put in the milk was rich in lactobacillus because curd has enough amount of lactobacillus. Now these lactobacillus got into the milk and they started converting the milk to curd. Okay, they started converting the milk to curd. So they grow in the milk and convert it to curd. Understood? Yes. During growth, the LAB produces acid that coagulate and partially digest the milk protein. Okay, casein is the milk protein, so that will be also digested. A small amount of curd added to fresh milk as inoculum or starter contains millions of LAB. So you can call this inoculum or you can call it starter. Now Tanuja, don't send starter emoji. Okay, which at suitable temperature multiply, thus converting milk to curd. Okay, which also improves its nutritional quality by increasing the vitamin B12 in our stomach too. So that's why people tell, when we go for gym and everything, they tell you can have yogurt. Okay, I think all of you would have heard this, no? Yogurt. So they tell Greek yogurt you can have. Why? Because again, it is rich in vitamin B12. Okay, and it makes sure that your stomach is clean or cleansed. Okay, or better. Okay, or better. Understood? Chalo. Okay, in our stomach too, the LAB plays a very beneficial role in checking disease causing microbes. So students, in the gut also there is 2 types of bacteria, one good bacteria, one bad bacteria. Okay, good bacteria, I thought you will send some chicken or something and you will be like, ma'am, starter. Okay, good bacteria and bad bacteria. Everywhere it's there. Okay, good bacteria, bad bacteria. So we have to increase the good bacteria in our stomach. Okay, we have to increase the good bacteria in our stomach. Yes, understood? Yes or no? Ha, she has come to class to make all of us hungry. Okay, I've come after 1 class. How can you do this to me? Okay, 2 classes I had today. So after 2 classes I've come here. This is my 3rd class for the day. Okay, so how can you do this, Tanuja? Sweetie Chauhan. Okay, the dough which is used for making food, okay, such as dosa and idli, is also fermented by bacteria, students. So there is a bacteria called as leuconostoc. Leuconostoc. Okay, this is going to be the ones. Chumma, your mom is not telling. Okay, I had one kuti lunch. Okay, students, your mom is not chumma telling that, okay, let's make the dosa batter 1 day early and, make the dosa next day morning. Why? Because this leuconostoc bacteria will be released and they will ferment this process. Okay, they will ferment this process. So if fermentation has to happen, you have to give the time period. No, you have to give at least like 8-9 hours so that the fermentation properly happens and there is a puffiness. Okay, there is a carbon dioxide build as well. That is how they make the dosa and idli. Okay, dosa and idli. Now the puffed up appearance of dough is due to the production of carbon dioxide gases. Can you tell which metabolic pathway is taking place in the result of carbon dioxide? So students, when normally glucose gets broken down, we are expecting what? C6H12O6. Okay, all of you go ask Baswa sir how glucose is getting broken down. Okay, glucose will get broken down to carbon dioxide and water. This is the normal thing. But in case this complete breakdown does not happen, you would have learned about lactic acid, no? Complete breakdown doesn't happen, you are going to get lactic acid. Okay, lactic acid. Okay, or you will get ethanol. Okay, 2 things you will get. Ethanol you will get. Okay, ethanol. That is your C6H5OH. Am I right or wrong, yeah? C6H5OH, no, ethanol. Okay, so you'll get two things. If glucose is not completely broken down, you will end up getting lactic acid or ethanol. So this happens with food also. Now what happens if complete breakdown doesn't happen? Immediately you will start getting, now how do we make wine? How do we make all these fermented drinks? That is because of this ethanol release only, right? Ethanol. Oh, it's right. Very good. Okay, ethanol release only. So that gives the alcoholic content. Understood? That gives the alcoholic content. Now, where do you think the bacteria of these fermentations come from? Similarly, the dough which is used for making bread is fermented using baker's yeast. I think all of you would have heard, no? When we go near the bakery, we get such good smell of the bread, students. Why? Because the bread has got puffy, the bread has got fluffy, carbon dioxide has got released, and that is the reason we get those fragrance. Okay, because when bread is getting roasted, like whatever the baking process, they add, before making the dough, they add the Saccharomyces cerevisiae. That is your baker's yeast. Baker's yeast is there, brewer's yeast is there, both same thing. Okay, just learn this only. Saccharomyces cerevisiae. A number of traditional drinks and foods are also made by fermentation by microbes called toddy. I think even in Telugu it is called, I think Tanuja, we tell kadla. Okay, in Malayalam it's called kadla. Kadla is actually, we take it from coconut trees and palm trees. Here they have mentioned only about palm trees, but nowadays it's done more in coconut trees because palm trees does not grow very easily. Okay, C2H6O. Nah, no, ethanol formula is not C2. Yeah, C2 ethanol. No, eth means two. Sorry. Oh, this is what? This is, sorry, this one. Both I erased off. C6H12O6. I've forgotten my chemistry students. C2H5OH. Yeah. All. Alcohol group. No, all. OH. Yeah. Kallu in Tamil also. Yeah, Tamil also. So it is a southern Indian drink which is fermented sap from palms. Okay, palms. So what they do is, students, they tie a pot. Okay, they tie a pot, and the sap that is coming from the plant will be deposited inside that. They will keep it for some period of time, and these will get fermented, and after fermentation this is going to release have a little alcoholic feel. Okay, before people used to like, all these alcohol wasn't this common, no? Like now, how many variations you have? You have beer, wine, gin, rum, other things, right? So these are, like, nowadays it's very common, but back then, in those days, let's say our great grandparents and all used to drink this toddy as a alcoholic drink, and it is not very alcoholic. Okay, it's like just to give that dizziness, they used to all drink, like it's just for like an occasion, like how people drink nowadays wine, the grape wine that they make at home, just like that. Okay, chalo, 1 second, students. Okay, now microbes are also used to ferment fish, soybean and bamboo shoots to make food. Cheese is one of the oldest food item in which microbes are used. Different varieties of cheese are known by the characteristic textures. Now you have feta cheese, you have the other sliced cheese, then you have shredder cheese, mozzarella cheese, and much more cheeses, right? Nowadays cheese only is more than the actual cheese. So that is a Swiss cheese. Okay, so that is because the texture. Okay, so texture means what, students? How much, see, the larger holes in Swiss cheese are due to the production of larger amount of carbon dioxide. So the, like, Tom and Jerry is not a joke. Okay, Tom and Jerry was real. Tom and Jerry, the cheese what they talk about is real. That was your Swiss cheese, like this. It was, no, it used to have bigger holes. Okay, it used to have bigger holes like this. So the larger holes indicate that this cheese has more carbon dioxide. Okay, the Swiss cheese had more carbon dioxide. Clear? Yes, the emoji cheese. The emoji cheese has also so many holes, no? Yeah, they had larger holes. So the larger holes indicated that there was larger amount of carbon dioxide, because the more you keep, carbon dioxide gives the puffiness. Okay, carbon dioxide gives the puffiness. Now, Roquefort cheese are ripened by growing a specific fungi on them, which gives them a particular flavor. Okay, so these are a little more expensive cheeses, which are, flavored in such a way. Okay, flavored in such a way Roquefort cheese. Okay, chalo. Yes. Tell now, ready? Yes, I do eat cheese. I really like cheese also. Okay. So in C, microbes in household. What and all we have? First we start with lab. Lactobacillus. Okay, lactobacillus bacteria. Lactobacillus bacteria is going to do the job of converting milk to curd. Okay, and this curd is going to act as the inoculum. Okay, inoculum which has a millions of bacteria in them. Okay, this is going to be a lab. Sorry. Next is going to be your leuconostoc, which is not mentioned in your NCERT, but you can remember leuconostoc, which is going to help with dosa and idli. And I think as South Indian, North Indians, everyone just loves this food. Yes or no? Just loves this food. Okay, so 1 second, students. Just give me 1 minute. Just 1 minute, okay. Okay, so sorry. Yeah, yes. So dosa idli, right? Dosa idli has, I think, more than, you know, South Indian food now. It has North Indians love it. Okay, North Indians love eating dosa and idli, and I think we all can relate to it. We have eaten from a very young age, but there maybe they do not have that easy availability like how it is available in the south. Okay, Bangalore, every nook and corner has a dosa center, dosa corner, wo that this everything is there. Okay, so dosa idli fermentation happens because of, okay, fermentation happens because of leuconostoc. Okay, leuconostoc. Did you all have your coffee? Okay. Next thing. Next is who, students? Your baker's yeast. Okay. So baker's yeast is going to be a normal yeast only. Okay. This is the same thing that will be used in your brewer's yeast as well. Okay, so Saccharomyces is Cerevisiae. Yeah, true. Now for us it has got boring because eating from a very small age, we have all got used to, you know, eating dosa idli so much that now we are like, oh my god, please don't make me eat an dosa again. Okay, but for them they get excited. Okay, they get very excited. Oh my god, dosa idli is there, and good dosa idli. Okay, so that's why making bread. Okay, done. Then what else? Toddy. See, nothing as such is mentioned, just remember the example toddy. Then remember Swiss cheese. You should know which are the examples where, you know, it is good for our household. Okay, and Roquefort cheese. It's actually Propionum roqueforti. So Propionum, Bacterium shermani, all these other, you know, bacteria that's going to help, but you do not have to know that toddy. I think I've already told my students. Okay, Roquefort cheese. These are the different things. Okay, these are the household help. That means microbes are not useless. They are actually helping us in some things. Okay, now microbes in industrial product. How are they helping us in building industry, students? So even in industry, microbes are used to synthesize a number of products valuable to human beings, like beverages. Is it valuable to us? Not sure about that, but it is valuable for some people, right? Beverages. Antibiotics are very, very valuable. So production of industrial scale requires growing microbes in very large vessels called as fermenters. So these microbes are grown in big, large tanks. Okay, big, large tanks called fermenters. Fermenters. So students, if you go to places, okay, if you go to places where they ferment beer, okay, where they make their own beers, there's a lot of places like in and around Bangalore, so it's a lot, but like if in your places, if you visit there, you will be able to see big steel fermenters. So what they do, they make their own beer there. So they are doing the process of fermentation. Okay, they're doing the process of fermentation. Okay, Otis. Hi Otis, Varun. So this fermentation happens. Okay, there are big steel containers where this fermentation will be. They'll be all connected with pipe. Those are breweries. That means they make their own beer. Okay, so they will wait for fermentation to happen. So all my bacha parties are here. Huh? How did all of them run and come at the same time? Microbes, especially yeast, have been used from time immemorial for the production of beverages. Okay, so yeast, that is your, okay, yes, please tell me. So here students, these who tell again your yeast, Saccharomyces cerevisiae only is the person who is going to be used as your brewer's yeast as well. Okay, brewer's yeast. Okay, Sony, why are you waiting for my class? Don't wait. I'm teaching no here. He's calling me too many times to Vedantu. Meet English. He's telling ma'am, please take more class. Okay, for this, the same yeast, Saccharomyces cerevisiae, used for bread making and commonly called as brewer's yeast. Okay, now is used for fermentation. Okay. So is used for fermenting malted cereals and fruit juices to produce ethanol. So what happens students, fermentation is done as I told you when glucose, okay, 12O6 does not gets completely broke down, we will get ethanol, C2H5OH. okay, we will get ethanol. Now this ethanol is a product of your incomplete fermentation or incomplete breakdown. Okay, so this ethanol is going to give, okay, this ethanol is going to give the feeling, the wine or whatever, the alcohol feeling. Okay, yes. Oh, he's only shocked. Oh, he did it, huh? Maybe Ratas is migrating to too many places, that's why. Full confusion he got. Learning too many things. Okay, so yes, look here. So what is going to happen students? Do you recollect metabolic reactions which result in the production of ethanol by yeast? Depending on the type of raw materials used for fermentation and the type of processing with or without distillation, different types of alcoholic drinks are obtained. Wine and beer are produced without distillation. Now here there is a catchy thing. Okay, here there is a catchy thing. What is the catchy thing here? The catchy thing is students, okay, the catchy thing is yeast can do fermentation. That we already got to know. Yeast can do fermentation. Since yeast is doing fermentation, it can do up to only 13%. Since after 13%, it will kill itself. Okay, it will kill itself because it will not be able to tolerate the alcohol content. Okay, yes or no? So look here, yeast can do fermentation. It can do up to how much? 13%, max to max 30%. By the time 13% alcohol or ethanol is there, yeast cannot survive. Yeast will die. Okay, yeast will die. Now what is going to happen students, because yeast is going to be able to make only 13%, beer is formed. Guys, how much did I give you the percentage? Yeah, go check your notes. Around 9% I guess. Okay, and wine up to 12%. Am I right? Just check and tell me. I have given you people the, okay, the, what is that, values. They'll be like, ma'am, which values? And you know, I have something very nice for all of you. I'll show you guys. Okay, I think you should all buy this. Yeah, these students. This is your notes. Okay, this is notes. Can you see? This is Vedantu's this book. Okay, yesterday I received it. So I don't know how many of you can notice. See, can you see? I'm bringing it closer. It's very nice. It's very nice. The entire booklet. So pretty. Okay, like so colorful. So this one I feel like studying from starting. Yes, flashcard book. Flashcards also are there. That also I have. Wait, I'll try to show you guys that also. This is flashcards. Flashcards also are very beautiful. Okay, see. This is like I'm going to read. 1 second, students. I don't want to mess it up. Yeah, see. Flashcards are here. Your cell chapter. So cell chapter. Everything is there. Anjali Van Leeuwenhoek, Robert Brown, your Scleden, Shawn, and cell theory. Okay, was Choff. See, everything is there. Very pretty, very pretty. Okay, if you have not invested anywhere, students, I think you should invest on this flashcards. Okay, because they're so pretty, you'll feel like studying. You know, the biggest problem that people don't study is because they see everything boring. Studies is boring, books are boring, no colors. Okay, Sony, I think currently it should be somewhere. You have to check in our channel, dear. There is a link, no? You have to check in that channel. Okay, the link I'll share it on Telegram. Okay, I don't know the exact price, but I'll share it on Telegram. Okay, chalo. So students, yeast fermentation. Okay, so students, check the percentage and just let me know. I had given all of you the percentage last time. So around 9% students, depends. Okay, beer to beer nowadays, I don't know what and all kinds of beers are there. So around 9% fermentation can happen to make the beer. Around 12% wine can be formed. Okay, around 12% wine can be formed. Okay, all of you, attendance. Exactly, the flashcards are so cute, Varun. Even I know. I feel like studying again. I think I might start studying again. Okay. Yes. So this is the percentage. So 13% when it reaches, the yeast will kill itself. Yeast will kill itself means yeast will not be able to survive. Now I need to make, let's say I need to make drinks which are more alcoholic, maybe up to 40%, okay, 60%. I have to make alcohol, you know, more alcoholic. So that time what I'm going to do, this is a not distillation. Okay. This is non-distillation process. That means without distillation, this is going to happen. Because why you want distillation? Yeast is doing fermentation. It can do up to 12%. So we will use yeast only. So without distillation. But for some alcoholic drinks, okay, the higher end drinks like your rum, whiskey, brandy and all of that, those people need more. Okay. Those people need more. Right, more we need to give like alcohol content, more alcohol content. So that's why we are going to see, whereas whiskey, brandy, rum are produced by distillation of the fermented broth. The photography of a fermentation plant is shown in your textbook. So students, the alcoholic drinks later, that is your rum, whiskey, brandy, gin, vodka, all of this are on a higher alcoholic level. That means it can go somewhere between 30 to 60% alcohol. Okay, 30 to 60% alcohol. If I want to make 30 to 60% alcohol, I cannot do it with the help of yeast. So for that, we are going to do distillation. Okay. So we are going to do distillation. So the question can come, which are the distillated, you know, alcohol, distillated less alcohol. So such a question will not come, but they'll ask you which are the alcohol that is, you know, made by distillation and which are the alcohol that is made by the fermentation of yeast. Okay, that you tell. Huh? No, I will. Why? What happened, Sony? Where am I going? I'm here only. Okay, so done. Now antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century and have, okay, wine is 9% to 12%. Okay, that's what I wrote, up to 12%, and have greatly contributed towards the welfare of human society. Anti, a Greek word means, okay. Yeah, I completed. Maybe I'll come less. Okay, I'll come less on the channel. Greek word that means against, or bio means life. Students, so against life. So antibiotic means what? Antibiotics. That means anti means against, not your auntie, okay? Against. And biotics means against life. Now, ma'am, against life means what, ma'am? Okay, against life means what? That means antigen is there. Okay, let's say a bad bacteria is there, that is life. So when we attack these people, we are going to attack with the help of antibiotics. So against life means what? We are killing another life. Understood? Okay, understood? Chalo, very good. Beer release lesser, 3 to 6%. I wrote here 9%. Up to 6%, I'll write, okay? Up to 6%. Okay, now, context of disease causing organisms. So, life, they are talking about the ones who will cause a problem. Now, let's say, okay, Himanshu, you have to do all the names. That is, like as I wrote, no, household. Then you have to do these, Staphylococci, Penicillium notatum. You have to do sewage treatment, very important, most important topic of this chapter, to be frank. Okay, then lot of names are there. This paragraph is very important. Where you have trichosporum, all that. Okay, monoscus type. So all that is there. Chalo. So antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of disease microbes. So students, it's not necessary that, like say now, let's say we take Dolo, we take any other antibiotics. Our thought process is not to kill the whole thing. Our thought process is to calm it down, right? Like when I'm having a viral fever, when I have a fungal infection, we use a medicine every time. It will not kill the microbe. It will sometimes just be like, bro, calm down, okay? So it is going to just tell calm down. So it will just retard the growth. It will just control the growth. Every time it's not going to kill. Okay, so antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of the disease causing microbes. Now, you are familiar with commonly used antibiotic called penicillium. So do you know that penicillium was the first antibiotic to be discovered, and it was chance discovery. So students, many chance discoveries has happened in biology. We should be proud. Okay, we should be proud. Alexander Fleming, while working on Staphylococci bacteria, once observed a mold growing in his unwashed culture plates around which Staphylococci could not grow. So students, very simple. We usually never do this. Okay, first of all, he is not supposed to do this. It's against the lab rules. So when you're entering into a lab, now when you go for MBBS, when you go for your masters, okay, good evening students, when you go for your masters, what happens? You will be allowed inside the lab. Microbiology lab is like, you know, very precious place. I'll tell you why precious place. Many people will be doing their PhD research there, their MSc research there, and they would have kept Petri plates. Okay, they would have kept Petri plates. Now, if you are a student who is careless and you leave an unwashed culture, that means inside that bacteria was growing, you forgot to wash it, you're supposed to dispose it properly, because these bacteria can become anytime. Okay, anytime. Exactly, he was lazy to do it, which he is not supposed to do it. Okay, like, you know, by mistake also we're not supposed to do. Why, students? Because see, imagine let's say Tanuja, Aman and Ratis is there in the lab, and let's say Aman is you know, having a fungal plate, Tanuja and Ratis is having a bacterial plate, which is good bacteria. Now by mistake, if Aman opens the fungal plate, okay, so we usually keep it upside down, like ulta we keep it, because spores will fall. Okay, spores will fall. So we do not keep it upside, like ulta. So what happens? Imagine by mistake Aman opens this, and the spores will spread in no minute. If the spores spread, it will go and grow on every other nutrient medium, because everything has components that will facilitate the growth. So just imagine, in 1 second you will damage the entire lab. All the microbiology Petri plates, every single place that is there will be filled with fungus growth. So this is actually not good to do. You should never leave an unwashed culture. And imagine in that unwashed culture, he had staphylococci, a good bacteria. Okay, good bacteria, matlab a strong bacteria. Okay, he found out that. So what happened is, wherever this penicillium was there, no, penicillium was making a zone of inhibition. We call it zone of inhibition. So when you're doing research, you'll understand, you'll have a scale to measure this. You'll measure the size. Okay, this zone of inhibition, students, this zone of inhibition was because here penicillium was growing and it was not allowing staphylococcus to invade into that place. That is when they understood that, okay, this is something that is an antibiotic which is consuming an antigen, or which is retarding the growth of antigen in the area where they are present. And understood? But will you ever do this in the lab? You should never ever do this in a lab. Okay, penicillium notatum. Okay, penicillium notatum. This was a bifluke experiment. Students, why students? This channel is from Vedantu only. Don't be lazy. Guys, don't be lazy. Okay, always wash your plates. Yes, now your parents will listen to the live and they'll be like, yes, we needed a teacher like this who will tell these monkeys to wash their own plate that they're eating. But I'm talking about Petri plate, okay? However, its full potential as an effective antibiotic was established much later by Ernest Chain and Flore. So Chain and Flore, we always say. This antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chain and Flore were awarded Nobel Prize in 1945. So because of his carelessness, he got a Nobel Prize. Okay, you will get 2 hittings. Okay, understood? Yes. So these 3 people, who is that? Chain and Flore. Okay, Chain and Flore, students, they use this antibiotic to get cure of soldiers during the World War. Okay, during the World War II. Clear? World War II. Chalo. Now, after penicillium, other antibiotics were purified from other microbes. Can you say, name some other antibiotic and find out their source? It's your homework, okay? Antibiotics have greatly improved our capacities to treat deadly diseases like plague, whooping cough, okay, diphtheria, leprosis, and which could kill millions of people. Many of it we got rid also. Now we have COVID. During the COVID time also, vaccines also has helped us, okay? But generally for most of the fevers, we give antibiotics as the first form of medicine until it's a viral infection. You do not have a choice. But otherwise, if it's a fungal, bacterial infection, we usually give antibiotics as the first stage of fighting. Okay, first stage of fighting. Now I think all of you, my students, already remember from human health that antibiotics, okay, that is, here we are talking about antibiotics. Same way in our body there is one person called antibodies. Yes or no? These antibodies are like the external antibiotics that we take, because these antibodies are going to fight straightforward. They're going to fight the people straightforward. Okay, yes, you people have homework. If you don't do homework, tomorrow I'll make you stand in the class. Okay, and you'll have to call your parents in the second day. Okay, yes. Now microbes are used for commercial and industrial production of certain chemicals like organic acids, alcohols, and enzymes. Okay, now students, this is a very important thing. Himanshu, if you are here, do not forget. Okay, this is also a very important thing. This is a very common NEET question. NTA's favorite question is this question. Okay, it's a favorite NTA's question. I'll tell you why. Because every year one word at least will come. Okay, so students, here, Aspergillus niger is going to form the citric acid. Okay, so acid producing fungus we are talking about. Okay. Yeah, tomorrow you will tell ma'am, my dog ate the book that I wrote the homework. Okay, my grandma ran away with the book, a rat ate the book. Tomorrow only all the lies you people will find and come. I know. Now look here. Acetobacter aceti, acetic acid. Okay, it forms the acetic acid. Okay, when we have students aspergillus niger, citric acid, acetobacter aceti. Very simple, no? Acetobacter aceti will form what? Acetic acid, aceti word is there, no? Clostridium butyrium, butyric acid. Okay, lactobacillus, lactic acid. The only thing that you have to remember. Okay, the only thing that you have to remember is going to be aspergillus niger because it has nothing connection with citric acid. Got it? Yes? Okay. Very good. Now students, yeast, Saccharomyces cerevisiae is used for, okay, it's used for commercial production of ethanol. Now, microbes are used for the production of enzymes. Lipases are used in detergent formulation. So, this is very important, okay? How many of you have thought this? That when I make the same apple juice in my home, okay, my apple juice will be whitish, okay, not even good looking. But the same thing if it is made in a store, okay, it's sold in a store, it will look like happy fizz. So that is students because of the use of pectinases and proteases, okay, like that one thing we have to study. So this four examples you remember, 1 2 3 4. Okay, here you have to remember lipases. Okay, lipases. I have tried so many times to make happy fizz in my home, and then I realized it's not even happy apple. Okay, so yes, students, because it's a joke, our lives are on a lie. Now next thing is, see, you must have noticed that the bottled fruit juices brought from the market are clearer. That means there is no fibers. Okay. If now normally if you make a mango milkshake at home, what exactly happens? Mango milkshake at home, if you make, you are going to get fibers. Okay, you are going to see sometimes the peel also will be there. So like it's very like it's not a very soothing feeling like how you drink maybe maza or how you drink another smoothie which is like made ready-made. Now what is the difference? The difference is this people use something called as a clarifier. Whom do they use? Clarifier. Okay. What are people sending my production house, you know? Okay, so these people use a clarifier. Okay, clarifier. Yeah, that is pectinase and protease. This is the reason why your juices that you buy from the market does not give you any feeling of like you're drinking a homemade something. Okay, yes. Now, streptococcus produced by bacterium streptococcus, streptokinase produced by streptococcus, sorry, streptococcus, modified by genetic engineering, is used as a clot buster. This is a very important question, okay? Streptokinase is used as a clot buster for removing clots. Now, what happens? Imagine a person has gone through a heart attack, or the doctor knows that this person in the previous or in the past had a, you know, mild attack, and it was because of the reason of having a block. So suddenly when the person faces a pain and they notice that it is because of a block, they will immediately give streptokinase. So when they give streptokinase, what exactly happens is this streptokinase will melt the clot. Now what do I mean by clot? All of you have studied body fluids and circulation. This is your blood vessel. Let's say this is the artery that is carrying the blood to the, you know, heart. Fat has got accumulated, okay, like this fat has got accumulated in the blood vessels, and because of which the person has gone through a heart attack. Now what is going to happen when we add streptokinase, students? This blood vessel, the clots are going to burst open without blocking, like they're going to melt down and just they'll just move, okay, they'll just move, okay, through the blood out, or they will remove it. Got it? So okay, so yes, wait, let me, class 10 abhi nahi. Okay, for removing clots from the blood vessels of the patient who have undergone myocardial infarction leading to heart attack. Understood? So clot buster we will use. Another bioactive molecule, okay, is cyclosporin A. What is it? Another bioactive molecule is cyclosporin A, that is used as an immunosuppressant agent. That is, imagine, okay, that is, imagine students, Tanuja has sold her kidney to buy iPhone, okay, and then they released iPhone 16 Pro. Now she wants to sell another kidney. So her mom will be like, okay, Tanuja, I'll give you my kidney, okay. All this is joke, okay, nothing she has done like that. So now what is going to happen when she takes the kidney from the mother? There is a chance that body will identify this kidney as a foreign particle, so the body will start rejecting it. Body doesn't know, no, it went for iPhone, so we cannot keep iPhone also in that kidney's place. So what we have to do, we have to get another kidney. So she'll be like, okay, fine, we will take the kidney. Okay, she's only shocked that my kidney is gone without even me knowing. So students, we cannot keep the 16 Pro here, no, and tell that please do osmoregulation, please help in RAS mechanism. We cannot tell. So we will get a kidney from somewhere. Okay, we'll get a kidney, and that kidney will, our body will start rejecting the kidney. So to make sure that the body does not get rejected, okay, the body does not reject this kidney, we'll have to, okay, we'll have to give immunosuppressive agent. Okay, when you give immunosuppressant agent, that is cyclosporin A, the body will calm down and the body will not reject the kidney immediately. Okay, yes, graft rejection, understood? Tanuja is shocked for life. Tanuja is like, ma'am, I will not come to your class. Yes or no? That you people fight, okay, you people fight and come to a conclusion whose kidney we should take, but we have already given cyclosporin A. Okay, cyclosporin A, so we have made students, so now our only job is to give. Okay, but you should decide whose kidney you are going to give and who is going to sell it for 16 Pro. Okay, you people fight and decide and let me know. Okay, so it's produced by a fungus, Tricho Shruti, is it, that's how you pronounce? Okay, so student, next one is produced, so this the cyclosporin A is formed by Trichoderma polyspore. Statins produced by yeast, that is Monoscus purpuris, okay, Monoscus purpuris, have been commercialized as blood cholesterol lowering agent. That means the statins that you get from produced by an yeast called Monoscus purpuris, it's actually understood or commercialized as a blood cholesterol lowering agents. Okay, yes, exactly. Your money is my money, my money is my money. So they are fighting over kidney. Okay, yes. Nay, Aman, I don't teach 10th. I am a NEET educator. I teach 11th, 12th and NEET. Okay, it acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. Clear or not? Now we have to write all this. So much we have to write. Ready? Chalo. So students, first thing is Aspergillus niger. Okay, Aspergillus niger, whatever you want to tell. Okay, it's going to give what? Citric acid. This is very important question. Okay, very important question. Citric acid. Next one, Acetobacter aceti. That is going to give acetic acid. Then next one, thank you, Shruti, if I'm pronouncing your name right. Butyric acid. Jaldi, one have to answer. Butyric acid by what butylatum? This people's kidney conversation will not take, I think it will take 2 days to settle down. Okay, this 2 students answer. I wrote 2 for you people. 2 you answer. Fast and furious. Only my students can do these things. Come on. Clostridium, think. Clostridium butylatum. Okay, Clostridium butylatum will come here and lactic acid will come here. Okay, Clostridium buty. Okay, and here what is going to happen? Lactic acid. Okay, lactic acid. Okay, yes, lactic acid. Okay, they don't know the spelling of this, so they're like, ma'am, lactic acid you catch. Very good, they know the spelling. Now otherwise I would have scolded you people. Okay, because I know my NEET students are so good at spelling. They have MCQs, so they don't even look at the spelling. Yes. Next is this one. You start with streptokinase. Okay, streptokinase is given by, okay, that streptokinase, just write the function. Okay, streptokinase is going to act as a clot buster. Clot buster. See, I know at least one student I understand. Streptokinase as clot buster. Then students, what else? Next one, immunosuppressant, that is cyclosporin A. Cyclosporin A, who is going to act as an immunosuppressant. Okay, immunosuppressant. Then you have statins. Please learn from where they are extracted. Okay, this one is from Monoscus purpuris. Statins are your cholesterol lowering agents. Lowering. Cholesterol lowering agents. Done? Yes or no? Sure. One second, students. You something I want to do. So many ads are playing. Okay, cholesterol lowering agent, clot buster, immunosuppressant, statins. Okay, done. Now, from where they are extracted, please make a note. Okay, this is Trichoderma polysporum, this is Monoscus purpuris, this is streptokinase. Okay, done. Now, this one also you can write. Staphylococcus was the how he found it. Penicillium notatum. Okay, Penicillium notatum. Okay, Penicillium notatum. Yes, done. Very good. Okay. Don't talk all this. Okay, Penicillium notatum. I'm so happy that I don't have tongue twisting after 3 classes. Penicillium notatum, done. That is going to be as antibiotics. Okay, antibiotics. Done. This much you remember, please. Especially this 4. This 4, if you forget, gone. Marks are gone. Okay, marks are gone. Microbes and sewage treatment. So students, here sewage treatment. Before I go for sewage treatment, let's go for a cootie break. Okay, where is the this one? Okay, all my students come back. Okay, so fast time went. Okay. Yeah. What and all you monkeys will do. Okay, come back. Okay, I'll see you guys. Hello students, I'm back. I hope all of you are also back. Let's start the class. So, oh, no. I asked you people genuinely, like, when do you want me to end? I did not ask like, sarcasm. I asked genuinely, when do you guys want me to end? Plan is till 8 o'clock. 8:10 max. Okay, that's the plan. So I asked like that. Okay, so students, what is sewage treatment plan? Wherever there's a lot of waste, right, these waste ultimately goes into the water bodies. Now we have to make sure that this water bodies that we are using for different purposes is purified from microbes, because it will be mixed with human excreta, it will be mixed with animal excreta, and these excreta will have a lot of unnecessary microbes. So we have to make sure that these are purified, because what happens, people at one point, they're just so careless, or they're very casual about the fact that these are all going into the water. Like when we litter, or especially in the village areas, they make the animals wash. Okay, they make people put their excreta, animal excreta, wash clothes in the same thing. Some people take bath in the same thing. So it's a mix, it's like a microbe, like a entire place where it's filled with microbes, right? So whenever we dispose any kind of water, okay, any kind of sewage water into the rivers, we have to go through a purification technique that is called as your sewage treatment plant. Okay, the treatment plan we use is STP, which is called sewage treatment plant. Okay, now how does this work? A major component of wastewater is human excreta. Okay, this municipal wastewater is also called as sewage. Now sewage is the one that runs through those sanitary tanks. Now if you see your road, maybe like when in one intersection, let's say this is one road where your house is there. Okay, this is your house. Okay, this road you will be able to see one sanitary tank like this. Okay, then from this road there will be one more connection. Okay, in another road there will be a sanitary tank. So inside the sanitary tank, what is there? Sewage water is there. That is the water that goes from our kitchen and washrooms. Okay, so these water we have to purify before sending it for to the rivers, so many of which are pathogenic. Have you ever wondered where this huge quantity of sewage or urban waste is disposed of daily? This cannot be discharged into the natural bodies like rivers and streams directly. You can understand why. Before disposal, hence sewage is treated in sewage treatment plants to make it less polluting. Treating wastewater is done by methods like parts by parts. Okay, first one is called primary treatment. Now see, since the waste material is coming from apartments, restaurants, okay, from houses, colonies, all of this, student, there'll be a lot of debris. Debris matlab like, say one of us ate lace, we threw it there. Now what happened? It was raining, this has gone and got into the sanitary tank. Or let's say cigarette, okay, cigarette, I wanted to tell, I don't know the word. Okay, we tell something for that. Cigarette, okay, that's after smoking, that person has gone and dumped it there. Okay, or due to rain, it has gone and settled somewhere. Okay, it has gone and settled somewhere. Now, in these cases, what is going to happen? Now, these debris have to be removed with the help of primary treatment. So, these treatment basically involves a physical removal. Okay, like students, now chai, when we make chai, what do? We remove the chai patti, we tell, no, like that tea leaves. We'll remove the tea leaves with physical remover. How? We will use a sieve. We'll use a sieve. Same way here also, let's say the content had a lace packet, okay, a lace packet, a chocolate cover, okay, a chocolate cover, wrapper, or a cigarette, okay, a cigarette. Now, with the help of a sieve, okay, with the help of a mesh or a sieve, we will do a primary treatment where we'll be able to remove all this. Understood? We will be able to remove all this. Now, so here, physical removal of particles, large and small, from the sewage through filtration and sedimentation. Okay, filtration and sedimentation. So the two process that you're going to do under physical removal is going to be what? Filtration and sedimentation. These are removed in stages. That is, initially floating debris is removed by sequential filtration. This one I told you. Then the soil. Like now, students, in sanitary tanks, What is this Tanuja sending? Okay, see, in sanitary tank, students, there will be a lot of soil, small pebbles, small rocks will be there. That I cannot remove through the sieve. Okay, even if I reduce the size of the sieve, it is still, soil is a very small particle. For that, we are going to do the second type of filtration. Okay, that is going to be removed by sedimentation. Sedimentation means what? I will just keep the, can't even see it, wait. Okay, I'll just keep the entire thing, okay, untouched for few hours. Automatically these particles will start settling in the bottom. These particles will start settling in the bottom. This is called as sedimentation. Sedimentation. Understood? Sequential filtration is then, sedimentation is there. All solids that settle from primary sludge and the supernatant forms the effluent. Primary sludge is whatever we have after settling, whatever we have got. Then the effluent from the, and the supernatant forms the effluent. So students, in a centrifuge tube also we always tell, no, pillet and supernatant, like that only. In this, whatever has settled, okay, whatever has settled, all the solid that has got settled is called the primary sludge. Okay, sludge. Whatever came on the top, okay, whatever came on the top, all this supernated, okay, all this supernated, this became the effluent. Now you tell me, will we use the primary sludge or the effluent? Do I need to filter the primary sludge or the effluent? Effluent, okay, very fluent, okay. Effluent, where should I take it? Should I take that or should I take the primary sludge? Definitely have to take the effluent, right? So the effluent, okay, the effluent from the primary settling tank is taken to the secondary treatment. Okay, it's taken to the secondary treatment. Now let's see what's the secondary treatment. Secondary treatment of or biological treatment, the primary effluent is passed into large aeration tank where it is constantly agitated mechanically and air is pumped into it. So students, continuously we keep agitating it. Means what? Agitation means what? Mixing it, okay, mechanically, and air is continuously pumped into it. This allows, so when air is coming, you imagine continuous oxygen supply we are doing. Continuous oxygen pumping we are doing. When we are going to do continuous oxygen pumping, what does it mean? It is going to allow aerobic bacteria to grow. Yes or no? Wherever, you know, organic matter is there, bacteria will go automatically. Okay, now since we are also providing air, that means oxygen also we are providing, that means aerobic bacteria will grow. Yes or no? So this allows vigorous growth of useful aerobic bacteria called flocks. What will you call that as? Flocks. Masses of bacteria associated with fungal filaments to form a mesh-like structure. Fungal filament means, or if you remember now, hyphae, mycelium, like the structures. Now, while growing, these microbes consume the major part of the organic matter. So, they will try to break down the organic matter in the effluent and try to consume it. Okay, this significantly reduces the BOD. So, less the BOD, okay, less the BOD, better is the water content. Okay, so the significant reduce in BOD, that is biochemical oxygen demand of the effluent. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. The sewage water is treated till the BOD reduce. That means if BOD reduce, what does it indicate? My water has less organic matter. If BOD is more, that means my water still has all the organic matter, nothing has gone. Okay, bacteria has not consumed it, not broken down it. Okay, so this bacteria has to oxidize it or break it down. So as BOD reduces, water purity also increases. Understood? Directly proportional. Okay, your dissolved oxygen is different story. Now the BOD test measures the rate of uptake of oxygen by microorganism in a sample of water. Indirectly, BOD is a measure of organic matter present in water. The greater the BOD of the wastewater, more it is polluting potential. Understood? More the BOD, more it is polluting. Okay, more it is polluting. Arthamaindi, yes or no? Understood. Yes. So how do I tell in Tamil? Did you understand? ಎಲ್ಲರಿಗೂ ಅರ್ಥ ಆಯ್ತಾ? That's how we tell in Kannada. In Tamil, Manasilayi. They're talking in Malayalam back to me. Tamil, what do you tell, yeah? Mm, Tamil, what do you tell? Oh my god, all the movies that I've seen is I've forgotten. Purinjicha. Purinjicha. Okay, Purinjada, Purinjicha. Okay, once the BOD of sewage or wastewater is reduced significantly, the effluent is then passed into settling tank. Okay, where the bacterial flocks are allowed to sediment. This sediment is called as activated sludge. So students, once the BOD of the wastewater is reduced, the effluent, we will pass it into another tank called as the settling tank. First it was aeration tank. In the aeration tank, we kept agitating it and we kept giving air. So flocks grew. That means aerobic bacteria grew. These aerobic bacteria kept reducing the BOD. Once the BOD reduced, we are going to the next step where we are adding this into a settling tank. Okay, in that we will let these people settle, and the settled one is called as activated sludge. Small, that means these bacteria will allow them to settle. Okay, the part of the activated sludge is pumped back. So now we have to use this activated sludge somewhere. That is, this bacteria also on a long run is going to be harmful. So I have to make sure that this bacteria is neither half of it is completely damaged and half of it I will take it for the aeration tank for my next process, because every time I'm not able to grow the bacteria. So a small portion of the activated sludge is pumped back into the aeration tank to serve as inoculum. The remaining part, okay, the remaining major part of the sludge is pumped into large tanks called anaerobic sludge digester. Students, obviously bacteria will be digested here. Okay, here other kinds of bacteria which grow anaerobically digest the bacteria and the fungi in the sludge. Pavam, their life is over. During this digestion, bacteria produces a mix of gas like methane, hydrogen sulfide, carbon dioxide. These gases form biogas and can be used as a source of energy as it is inflammable. Okay, so this biogas is the one that we make all this fuels and all in the rural areas. Okay, so is this clear? Clear? Not clear? So students, first step, okay, there are two types of treatment. Okay, physical treatment, primary and secondary. Uski niche we operate physical. Oh my god, I don't know how many languages am I talking in 1 minute. Primary treatment and secondary treatment. Primary and secondary. Now under primary, you are going to have two steps. Okay, one is called filtration, that is called sequential filtration, and one is called sedimentation. One is called sequential filtration. This is like a physical method where we will remove the what have they given here? Have they used the word debris? No. Remove the floating debris. No? Yes, floating debris. Okay, remove the floating debris. Okay, next is going to be sedimentation. So here students, we got all of this with the help of sequential filtration. Next is going to be sedimentation, where you are going to allow this to settle down. Okay, so we are going to allow, this is for the grits and debris. Okay, this is small particles, soil, all of this we are going to, with the help of sedimentation, we are going to take it. So now this is the mixture that I have. Obviously this is not being such a small dabba. Okay, I am drawing it like that because obviously I cannot draw the whole thing. This is going to be the soil, grits, debris, everything is going to be here. Top of that you are going to have the supernatant. Okay, yes. Now going back, this portion, this is called your primary sludge. Okay, primary sludge, and this is going to be your effluent. Okay, effluent. So this is the effluent. Yes. Now this effluent is going to be transferred to your secondary treatment into the aeration tank. Into what? Your aeration tank. in the aeration tank. Okay, this is the only thing, only main topic from this chapter. Others are all terminology. See, I'm not telling that only this you have to learn. I'm telling that this is one of the most important. Okay, that you cannot skip. You cannot skip it by any means. Okay, done. Now, students, aeration tank. In the aeration, what happened here? Yeah. In the aeration tank, continuous agitation. You get no agitated when you're not able to do well. So, like the agitation and continuous air pumping. Air will be pumped. This will lead to the growth of flocs. Okay, these are what, students? Aerobic bacteria. Okay, aerobic bacteria. Now what are they going to do? They are going to consume. Okay, they are going to consume the organic matter. I'm writing organic matter. Okay, flocs. What spelling? FLOCS it is. Okay, consume the organic matter and they are going to reduce the BOD. Okay, they are going to reduce the BOD. Now that BOD, okay, once the BOD is reduced, right, once the BOD is reduced, that water, okay, that water will be transferred into your next tank. What is the next tank? Same sound, ma'am, it seems. Wow, wow, wow. So instead of hair, I'm telling you, you have a lot of hair. I'm writing HAIR. No, I'm writing HARE. Does that mean you have a lot of hair? In the biodata that you are going to get married, I'm going to write he has a lot of hair. Okay, so it will be HARE. So the girl will reject you. She's not going to think, no, that you have this hair. She'll think, okay, fine, you're bald, only hair he has, so many rabbits he has. What am I supposed to do with that? So your marriage is gone. Okay, over for you. So same sound means this also you assume the same. For everything I'll have an answer. That is what my students are thinking. They'll be like, ma'am, whatever we tell you, you will have an answer for it. Okay, so BOD will reduce, students, so this water will be taken to sedimentation. Means like, I surrender. Sedimentation tank. Sedimentation tank. No, settling tank. No, why am I writing sedimentation? You people are not telling also. Settling tank. We're settling the issue. Okay, settling tank. So now from settling tank, what is going to happen? We have to digest that flocs which was there, no? So this flocs will have to be digested. For that we are going to use an anaerobic sludge digester. Anaerobic sludge digester. Okay, now this anaerobic sludge digester is going to digest it, but before that I am going to take, okay, there is going to be a portion of the activated sludge. That means I'm going to let them settle. Okay, activated sludge. Now from ma'am, where did I get activated sludge? I got it from your textbook. Okay, now let's say this is the tank. Okay, this is the settling tank. In this, what did I do? I let them sediment. After they, BOD was reduced, I let them sediment. Okay, then look here. So here the sediment, this is the bacteria, because the bacteria has consumed all the organic matter. Now the bacteria is going to settle. This is going to be my activated sludge. Understood? This is going to be, if you understand this connection, no, then there is nothing difficult. This activated sludge, students, small portion, okay, a small portion will go into my aeration tank. Okay, small portion will go into the aeration tank. Why? For the next process to start there. Aeration tank, what is going to happen? Consuming the organic matter is going to happen. Okay, consuming the organic matter is going to happen. So after that, the rest of the flocs, okay, or let's say activated sludge, okay, I'll remove the word this. I'll write activated sludge only. There's no confusion as well. Activated sludge. Okay, here I'm going to write it is bacteria. Okay, bacteria. Activated sludge. The rest of it is going to be digested by anaerobic sludge digester, where there will be anaerobic bacteria. Students, Kattappa story, students, Kattappa. Not Kattappa, no, Bahubali. Okay, after Bahubali was used for all the wars, he's been killed in anaerobic sludge digester. Like that only, the flocks have been killed in anaerobic sludge digester. Okay, chalo. Now this is done. Now students, what is going to happen after this? Definitely the BOD will reduce, right? Now the water is more in a better situation. Now for after this, you can send the same thing for tertiary. Okay, tertiary treatment, that is for making this drinkable. Okay, tertiary treatment to let this water, use it for drinking purpose. Okay, drinking purpose. Clear? Yes or no? Okay. So here they have not told, but here you can just see. Okay, biogas. Now see, aerobic, anaerobic sludge digesting. Here other kinds of bacteria which grow anaerobically digest the bacteria and fungi. During this digestion, bacteria produces a mixture of gas. This gas thing is the your biogas. Okay, gas will be formed. Which are the gas? Methane. Okay, hydrogen sulfide H2S, and your carbon dioxide. Okay, carbon dioxide. These three will be formed. These three will be used for biogas. This tree will be used for biogas. Yes, got it? Done? Understood? Done. Okay, now the Ministry of Environment. Okay, the Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of, okay, that is, we have to eat food also, da. You tell Papa Cooker to keep quiet, how will life happen? Okay, sewage treatment plan. Wasn't that audible? The Ministry of Environment. Okay, the Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of, okay. Hey, Kutti sound only game. Chumma don't do. This will become which are gas contain. Hey, Kutti sound, what full volume you have less? After class, you will be like, Tata Baba, ma'am, see you next time. Wow. After that, who will go and eat? Right? Glucose I need now to take tomorrow's class. So only treated sewage may be discharged in the rivers. A visit to a sewage treatment plant situated in any place near you will be very interesting and educating express. Go learn something. Okay? Go learn something. Ganga Action Plan. Okay, Ganga Action Plan and Yamuna Action Plan. Yamuna Action Plan. Got it? These two plans were done to save the rivers. Students, it's very, very, sad to see the rivers. That was so beautiful. Okay, that was so beautiful once. Now it is so sad to see them. Chalo, last topic, production of biogas. Okay. Production of biogas. Okay, so production of biogas is very simple. Now already you got to know, no, that these gases will be released. So students, cow dung. Okay, so first thing you have to understand, bacteria is there. Archaebacteria. This archaebacteria has a bacteria called methanogens. Okay. This methanogens release methane. This methanogens release what? Methane. This methane, okay. Now where is this methanogens present? In the animal, okay, in ruminant animals. ruminant animals. Okay, in ruminant animals, these ruminant animals are who? Your cow and all the cattle organisms. Okay, same thing, cow. So this ruminant animal's gut will have this methane producing methanogens. Now students, when the cow dung is formed, okay, when the cow dung is formed, this cow dung will be rich in methane gas, and this methane gas is what we need for the production of biogas. So we'll take this cow dung, okay, we will add it here, we'll make a slurry. Okay, this cow dung plus water, we will make it a slurry. Okay, we'll make it a slurry, and this slurry will be added here. Now from there the gases will come, okay, gases will come and they will get settled here. Okay, they will get settled here, and these people, there's a holder, okay, gas holder. So when the gas is coming, what is going to happen? Gas is definitely going to exert pressure, no, partial pressure, you have learned, no. So it's going to move, the gas holder is going to dance like this. Okay, like this, it's dancing. That time we'll get to know gases. Then the gas will be taken, collected from here, okay, and this will lead to the production of biogas. Understood? Anyway, people are shocked. Yes. Okay, so slurry, that is cow dung plus Niru, Pani, okay, together we will, this is the dung, okay. To this the gases will be collected. Which are the gases? CH4, CO2, hydrogen sulfide, okay, all these gases will be collected. These gases will be used as biogas, okay, and this biogas will be used as a fuel. Fuel meaning for burning and all of that. Okay, yes, I can see it. Very pro dancer. Okay, Max Pro. So students, microbes as biocontrol agents. Now biotechnology people learned, so it is baby for all of you. Okay. Now biocontrol refers to the use of biological method, okay, for controlling plant disease. Okay, plant disease. I'm searching for my phone. I'm like, where is my phone? I'm seeing the live there only, but I forgot where is my phone. Have it happened to you? Specs is on your head and then you ask everyone where's the specs. One second students. One second. Okay. Where is Gopika ma'am? That's a good joke. It was a good joke. Exactly, many times. Okay, I'm searching. I'm like, oh my god, I haven't seen my phone in so many times, so much time. Where's my phone? Where's my phone? Then I realized my phone only. I'm looking the live, no. So yes. 1 second, students. Let me just connect this. So much hard work. Yeah. No chat. I cannot see your chats. Okay, chalo. So students, all of you like here conjuice. Such conjuice in every channel. Okay, in modern society, these, so students, biological methods, how? Like instead of putting so many fertilizers and other things, better ways to use biocontrol agent. That is the example of your BT cotton, Bacillus thuringiensis. This Bacillus thuringiensis is an example that it will only attack the let's say the caterpillars that come and feed on the cotton. Okay, or the lepidopteran. So they will only attack some types of animals. They are very specific. But that is not the case in the case of fertilizers or insecticide. They will attack in the mass. Whatever is useful, whatever is not useful, everything. Okay, everything will get damaged. Where have we gone, no? So these problems have been tackled increasingly by the use of chemicals, by the use of insecticide and pesticide. These chemicals are toxic and extremely harmful to human beings and animals alike. Have been polluting our environment, soil, ground, water, everything, students. Now we know, no, that the amount of fertilizers that we use, we are only consuming it. It's like we are only putting, we are only consuming, okay? And people are not understanding. Okay, people are not understanding. So I told you, last time when I was teaching you biotechnology, to make the leaf, the coriander leaf looks like this, no? They have a juvenile face and all of that, like this. Coriander leaf looks like this. To make the coriander leaf look like this, they are adding some extremely harmful, like some magnesium oxalate or something, to make it look like this. So by the time it comes in our home, it will look like this. Okay, it will look like something like this. Yes or no? Have you all noticed? It will look like this. But to make it look like this, so that it's exporting value. Okay, exporting value means, I'm not telling exporting, importing outside, but exporting to the markets. Okay, to reach up to the market, its value is going to increase. Okay, and places like all this 10 minutes delivery, no, students? Okay, 10 minutes delivery is there. This 10 minutes delivery, people also show such images or wants people to buy it because of how fresh it looks. Yes or no? Now, since it has to look so fresh, they are like peer pressure. Okay, like that vegetable pressure they have. So they will try to make it look very this one. But the problem is, by the time we consume it, we will not live more than 40 years if we are continuously going to consume this. But there is no other option at this point, because everyone tells that eat healthy. Now, eat healthy means if it is coming from the market itself like this, how can we eat healthy? Okay, chalo. Now, biological control of pest and disease. In agriculture, there is a method of controlling pests that relies on natural predation rather than introduced chemical. A key belief of organic farmers is that biodiversity further health. The more variety a landscape has, the more sustainability it is. The organic farmer therefore works to create a system where the insects that are sometimes called pests are not eradicated, but instead are kept manageable levels. That means, students, see, it's easy to kill something, right? It's easy to kill something. Let's say there's a frog. Drawing this frog is a task. Okay, there is a frog. There's a frog. Now this frog is going to be eaten by a snake. Okay, a snake is going to eat it. This frog has eaten a fly. Okay, it's eaten a fly. This fly has fed on some plant. Okay, some plant. Got it? Now, okay, now if I kill something, let's say I'm killing this, okay, this dragonfly or whatever, okay, this dragonfly or whatever. I have water, thank you so much. Okay, I'm going to kill this dragonfly or anything. Let's say this fly completely. I'm going to eradicate it completely because it's feeding on this plant. Okay, feeding on a plant that I'm going to commercialize or I am going to sell. Now, if I eradicate this from the ecosystem, don't you think the entire food chain is going to be harmed? The entire biodiversity is going to be harmed. So they are like, let's not eradicate it. Let's just reduce its effect on our plants. Understood? So eradication, better than eradication, is they doing this to just minimize, okay, reduce the effect on the plants. Paka, okay? Why is Sony shocked seeing my frog? And harmful life forms. So in the use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticide. An important part of biological farming approach is to become familiar with the various life forms that inhabit the field, predators as well as the pests and their life cycle. So as I told you, that you have to be aware. You have to be more aware that, oh my god, I am going to damage this because okay, if I am going to be damaging an entire organism whose ecosystem, like, which has a role in the food chain. Okay, since it has a role in the food chain, students, we cannot damage it. Understand? No, we cannot damage it. So since we cannot damage it, we have to think of a method where we control these organisms. Okay? People are so funny. Okay, done. Go home. No, it's not done. Wait. Okay, they are very familiar beetle with red and black markings. That is your ladybird. Okay, ladybird. The ladybird and the dragonflies are useful to get rid of aphids and mosquitoes. Okay, respectively. So here you have to remember who is going to eradicate whom. Okay, in the exam, don't write mosquitoes will eradicate ladybird. Dragonflies are used to get rid of aphids and mosquitoes. Okay, mosquitoes. Now, an example of microbial biocontrol agents that can be introduced in order to control butterfly caterpillars is the bacteria called Bacillus thuringiensis. So students, for those students who do not know what is Bacillus thuringiensis, go watch my biotechnology video. Okay, so yes, so you can just put my name and you can just search the chapter that you want, you will get the videos. Okay, yes. So students, here what is going to happen? Bacillus thuringiensis is a bacteria that is releasing something called as the Bt toxin for its protection. This Bt toxin is in the inactivated form, so we will buy the spores of it and we will add it into the cotton, or let's say anything for that matter, lady's finger, tomato, we will just spread it there. Okay, the spores, we'll mix it with water and we will spread it. Now what is going to happen, student? This is very specific. That means it will only get activated. So much is there. Okay, it will only get activated when what is there? When there is an alkaline pH. When there is a alkaline pH. Our stomach has what pH? We have acidic pH. So this will not get activated in our stomach. No. So when we add this Bt spores, okay, Bt spores we tell this, okay, Bt toxin or Bt spores, which is mixed with water, we spread it on cotton plant. Okay, this is the cotton plant. This is a cotton plant. We spray it on them. Now what is going to happen? This will attack, or this will get activated in the caterpillar gut. Okay, it will get activated in the caterpillar gut and it will kill the caterpillar. But will it kill something else? It will not kill something else. Yes? I don't have names, students. So many names are there that now I don't have many. Don't know which is my actual name. Done? Done? Okay, it will get activated in the caterpillar's gut. Why? Because in the caterpillar's gut, there is alkaline pH. Alkaline pH. Done, done. Chalo. That is your biocontrol. That is your biocontrol. So they are available in sachets, whatever I told you, same thing. Bacillus thuringiensis is the full form. Bt cotton is one of the examples, but now we are using in tomato, lady's finger and all of that. Okay. Now a biological control being developed for the use when, this is extra information. See, a biological control being developed for the use in treatment of plant disease is fungus trichoderma. Trichoderma species of free-living fungi that are very common in the root ecosystem. They are effective biocontrol agents for several pathogens. So students, even though they are living, okay, okay, even though they are living in on the roots of the plant, they are not harming the roots of the plant. They are helping in the roots to become better because they are not going to let organisms to come and attack on the roots. Understood? Now, baculovirus are pathogens that attack insects and other arthropods. So baculovirus is an pathogen that will attack insects and other arthropods. The majority of baculovirus used as biocontrol agents are nucleopolyhedrovirus. Okay, nucleopolyhedrovirus. These viruses are excellent candidates and they have a very specific narrow spectrum. This is an important point. Narrow spectrum insecticidal applications. Understood? Yes or no? Okay, narrow spectrum insecticidal application. They have been shown to have no negative impact on the plants, mammals, birds, fishes, or even on non-target insects. This is especially desirable when beneficial insects are being considered to aid in overall integrated pest management, that is IPM program, or even an ecological sensitive areas being treated. So this you just remember, trichoderma baculovirus, okay, and nucleopolyhedrovirus. These two terms you remember. Now students, organic farming, that is those where we use biofertilizers instead of very poisonous fertilizers. We use biofertilizers, okay. So when we use biofertilizers, what exactly happens is it's not going to harm us when we consume it. Okay, it's not going to harm us when we consume it. Okay, and one symbiotic relationship is rhizobium. I think all of you know this, no? Rhizobium, that is students, on the roots of the leguminous plants. What is leguminous plants? These are the people who will be able to fix the atmospheric nitrogen and convert it into ammonia, other things, and increase the fertility of the soil. So in paddy field, you know, now if I was a farmer, okay, there is a paddy field. In between the paddy, I would have kept leguminous plants like pea or beans. Why? Because these people will be able to take the atmospheric nitrogen, convert it into ammonia, and increase the soil fertility. So these plants have an symbiotic. Symbiotic means what? Happy relationship. Okay, with whom? Rhizobium. Okay, with whom? Rhizobium. So these bacteria fix atmospheric nitrogen into organic forms, which is used by the plants as a nutrients. Understood? Yes or no? So then students, even in higher organism, there is something called as mycorrhiza. Okay, mycorrhiza, especially seen by globus. Okay, shown by globus. So these people also live in pinus plant and all of that. They are very tall trees, no? So these people will help in converting, recycling some sulfurs, you know, magnesium, some minerals. Okay, some minerals. Yes. So other bacteria can fix atmospheric nitrogen while free living in the soil. Example, azospirillum and azotobacter, thus enriching nitrogen content. So this is also two examples. Okay, students, examples. This chapter is full of examples. You have to remember the examples or else gone. Okay. This is mycorrhiza, I told you. See, fungi also known to have symbiotic relationship with plants, that is called mycorrhiza. Many members of genus Glomus form mycorrhiza. So they are the main people who are going to have the symbiotic relationship. Every fungus cannot capably do this. Okay. The fungal symbionts in this association absorb phosphorus from the soil and passes it to the plant. So students, in tall plants, what exactly happens? These are the problems that happen, so that they might not get the nutrient that is enough. So when in such situation, these people will be able to help them. Okay, done. Now, cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments, many of which can fix atmospheric nitrogen. Okay, many of them who can fix atmospheric nitrogen, like Anabena, Nostoc, Oscillatoria. Okay, so, to be frank, Oscillatoria does not have heterocyst. Remember, heterocyst cells will be able to fix nitrogen. Actually, this research had happened. And before in practical, students, I don't know if you remember, in your practicals we used to write this, that you know Oscillatoria has heterocyst, but later research proved that they do not have heterocyst. Okay, so this is a debatable topic. This heterocyst is the one who is going to do nitrogen fixation. Okay, nitrogen fixation. So in paddy fields, cyanobacteria serves as an important biofertilizers. Blue-green algae also add organic matter to the soil, increases its fertility. Currently in our country, a number of biofertilizers are available commercially in the market, and farmers use these regularly in their fields to replenish the soil nutrients. Okay, to increase the soil nutrients. Done, all of you? Okay, names you have to remember. Okay, very simple names are there. Rhizobium. Okay, with leguminous plants. Legumes also you can write. Okay, then mycorrhiza, with pinus plant actually, or you can write higher plants. Okay, higher plants. Then here you can write Anabena. Students, don't write Annabella. Okay, Nostoc, Oscillatoria, okay, Azotobacter, Spirillum, All these are for, again, nitrogen fixation. Done. Nitrogen fixation. That is your puri kahani of your microbes chapter. 2 hours microbes chapter is done. Okay, this is all you need to know. Just write the names wherever I have written like this. Okay, in between I have written notes. No, that notes you people please kindly try to copy it up. Okay, done, dusted, happy. Okay, so yes students, thank you so much for joining. So we have completed our microbes chapter. So if you have any doubts, do not let me know in the comments. Okay, my tongue is twisting. These people prayed for it and right now it's happening. Okay, so yes, see you all in the next class with another chapter by itself. So we have completed our NCERT line by line of microbes chapter. So that's it. Thank you so much for joining. See you all in the next class. Anything to say, students? These people will tell ma'am didn't tell bye and she left. Okay, thank you. Yes, microbes khatam, pura khatam. So happy, no? Tongue got twisted. So mean you people are growing up to be. Yes or no? Yes or no? Okay. Chalo, see you guys in next class.